Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Last video we were talking about mitosis. This week we're jumping into meiosis. As always, we take these first few moments of the video to allow you to grab those terms and definition slides that hold all the important vocabulary we're going to be using in the video today. As always, I'm also going to remind you that if you would like the PDF version of my slides for absolutely free, you can head on over to my Patreon page. The link is down in the description below and grab that PDF. A quick reminder though, if you are a patron, you do get early access to the Biology for Beginner videos and any study material that goes with them at least a day early, as well as early access to other videos based on our upload schedule and if they actually get done in time for that. So without further ado, let's get into meiosis. Before we get into the nitty-gritty of meiosis, let's pause and talk a little bit about cells and chromosomes. So the first thing we're going to talk about here is what is known as somatic cells. Now, a somatic cell is basically just your typical cell within a multicellular organism. In general, they're going to be diploid or 2N, and basically diploid just means that they have two copies of the chromosomes. And this is going to make up the majority of mammals and plants. Keep in mind, however, that there are exceptions to diploid. Diploid is not necessarily the rule, but biology is weird, and this is a beginner bio video, so for our purposes, diploid, 2N, that's the norm. That's all that you need to know. And they contain a pair of homologous chromosomes, one from mom and one from dad. The homologous chromosomes are essentially chromosomes that carry pretty much the same information, but they might have different versions of that information. Now, on these homologous chromosomes, you get the genes, and each gene can be found on a chromosome at a particular location known as a locus. And again, the homologous chromosomes have the coding for the same gene at the same locus, but they may have different versions of that gene. So let's talk a little bit about these chromosomes. Now, there are two different types of chromosomes. Chromosomes that determine biological sex, and these chromosomes are called sex chromosomes. So for example, in humans, we have that X and Y chromosome. In different organisms, it is different, but humans, X and Y. And then all the other chromosomes are called autosomes. And now they just kind of code for everything else. They hold all the other genes that are necessary for life to do the life thing. So a quick little recap. The somatic cells pretty much are the cells that hold everything. They're just your normal cell. In general, they're going to be diploid or 2N, and that 2N means that you have two copies of every single chromosome. For humans, we have 46 chromosomes in a normal somatic cell, so diploid for us is 2N. And they're going to mean that there's 23 unique chromosomes and a copy of each of those, so the homologous chromosome. So that 46 chromosomes is made of 23 different chromosomes and their homologous chromosome. Of homologous chromosomes, you get one from mom and one from dad. On that chromosome, all of the genes are going to occur at the same gene, same locus on each chromosome. However, it may or may not be a slightly different version of that gene. We'll get a little more into different versions of genes in future chapters when we actually begin to discuss genetics in a little more depth. Now that we've talked about somatic cells, let's talk about gametes, because that's where things begin to get a little weird. Now, gametes are your egg and sperm cells. So they're the cells that combine to make the unique new organism through sexual reproduction. and gametes are going to be haploid, or just N. And what this means is that you have one set of chromosomes. So just one chromosome, you no longer have that second homologous chromosome in a gamete. It's just N. So in humans, you have 23 chromosomes per gamete. So you have 22 autosomes and one sex chromosome. So the 22 autosomes that pretty much code for everything, and the one sex chromosome that goes towards determining the biological sex of the future offspring. So in women, they only have X chromosomes. So it'd be 22 autosomes and an X chromosome in those gametes. And then in the male sperm cell, you have the 22 autosomes and then either an X or a Y. And that's just because we only have the 23 haploid condition of the sex chromosome, or excuse me, of the gametes. And then with those egg and sperm cells, you get fertilization, which is where the nucleus of a sperm cell fuses with the nucleus of an egg cell. And then that sort of fused mass of those two cells is what is known as a zygote. And the zygote is 2N, so it's diploid. So in a normal kind of life cycle of a human that you can kind of see in the diagram here on the slide, in the blue color-coded area, from fertilization to that zygote, that kind of first step in the life cycle, is the beginning of the diploid 2N period. Now, throughout all the rest of mitosis and development up until adulthood, those gametes are going to be produced, where you get that haploid sort of area with the sperm and the egg cells. And then when a sperm nucleus fuses with an egg nucleus, you get that fertilization, and it begins again with the diploid zygote. So how exactly does a 2N diploid organism have cells go to that haploid state? Well, they get from that diploid parent cell to the haploid daughter cells through a process known as meiosis. And it's very similar to what we discussed in the previous video with mitosis, but it has a few extra steps. Now, if you haven't watched the mitosis video yet, I highly recommend you go back and do so, because there are going to be things that we're going to gloss over in this video that were discussed in more depth in the last one. So go ahead, click off of this, go watch mitosis. I can wait. Welcome back to this video now that you've caught up on mitosis. The few extra steps of meiosis that we don't see in mitosis are basically going to be the two parts, and one of those parts has a little bit more mixed into it. So it's meiosis 1 and meiosis 2, because scientists are clever and came up with clever names. But first, before a cell can get into meiosis, there's interphase. Before any kind of division or anything happens in any cell, you're going to have interphase. And again, just a little recap, interphase is where normal cell function happens and all that lovely DNA duplication happens. If you go back to the mitosis video, there is a little more in-depth explanation of what interphase is, but because that was explained in the last video, this is all we're mentioning on interphase here. So meiosis 1. Meiosis 1 is basically where all the differences between meiosis and mitosis are. During meiosis 1, you get a thing known as crossing over that occurs, and this is where you start with one diploid parent cell and you end with two haploid daughter cells. However, in those haploid daughter cells, the sister chromatids are still attached to one another. They have not been separated yet. So the first step in meiosis 1 is going to be prophase 1. Again, scientists are super clever and come up with super clever names here. So basically what happens in prophase 1, you have these homologous chromosomes that are going to be made up of those sister chromatids. They're going to come together as pairs. and form those chromosome tetrads. And just think of it as tetrad because you have 4 points. The chromosomes are these little X's, and then you have the 4 points because you have those 4 noodly arms there. And this is where the crossing over is going to occur. We will get a little more in depth on crossing over in future slides, so just sit tight here and just understand that it occurs during prophase 1. And also during this prophase 1, the chromosomes are going to coil tightly. You're going to have that nuclear envelope breakdown, and the spindle fibers are going to form and attach to those chromosomes. So very similar to prophase and prometaphase of mitosis here. After prophase 1, we get metaphase 1. And during metaphase 1, those chromosome tetrads are going to line up at that metaphase plate. And the homologous chromosomes are being held together at those sites of crossing over. And then 1 spindle fiber per homologous chromosome is going to attach. So you'll notice each homologous chromosome has its own spindle fiber, as opposed to each sister chromatid having its own spindle fiber. At this point, the sister chromatids are going to remain attached. And then after this, we have anaphase 1, where the chromosome tetrads are split, and the homologous chromosomes move towards opposite poles of that cell. And again, keep in mind that the sister chromatids are still remaining attached at this point. It's just those homologous chromosomes being pulled apart. And then we get the telophase 1, where the chromosomes reach those opposite poles of the cell and cytokinesis occurs. So the cytoplasm divides and the cell splits in 2. And what you end up with are 2 haploid daughter cells. And in these haploid daughter cells, those sister chromatids are still attached to one another. They still have not been separated. It just happens to be the homologous chromosomes have been separated. So at this point, if we're talking about human meiosis, you have now gone from 46 chromosomes per cell to 23 chromosomes per cell. But we still have the duplication of that DNA there because the sister chromatids are still attached. So we need to undergo one more process of splitting. But before that happens, there may or may not be a second period of interphase. But if there is a second period of interphase, there's going to be no more DNA replication. It's just kind of a period prior to meiosis 2, where the cell prepares for meiosis 2. We're not going to go super in depth on meiosis 2 because it's basically exactly the same as mitosis, except that you begin with 2 haploid cells and you end with 4 haploid cells. And there was no DNA replication immediately prior to this, but you still get that prophase 2 where the spindle fibers form and are going to attach to those chromosomes that are floating around in those 2 haploid cells. You still get that metaphase 2 where they're going to line up at the middle, and then the anaphase 2 where the sister chromatids are pulled apart. And then the telophase 2 and cytokinesis where those cells are split and the nuclei form. But again, if you want a little more in-depth of exactly what's going on during the prophase, metaphase, anaphase, telophase, please go back to the mitosis video, because meiosis 2, almost exactly the same, except with the whole haploid versus diploid thing. So meiosis ends up with random combinations. And that is one of the big deals you need to remember about meiosis. And one of the things that is so important to life that experiences the sexual reproduction. So here we have an image from the book showing 2 equally probable arrangements of where the chromosomes are going to end up during meiosis. In possibility A, all of the red chromosomes end up together and all of the blue chromosomes end up together. Probability 2, you end up with a split where each brand new cell at the end of meiosis has 1 blue and 1 red. Again, I would like to stress that that is equally probable. And in this situation, it's likely that there are more probabilities as well. But the way that they have done it, there are only 4 chromosomes, so that does limit the possibilities. But when you're talking about an organism that has 26 chromosomes, the possibilities of the random combinations of those chromosomes only goes up. One thing that increases this random combination of chromosomes and genes is this process known as crossing over. Now, during crossing over, it's important to remember that we have homologous chromosomes that may carry different versions of genes. And so these homologous chromosomes may trade corresponding segments. And this trading will occur at a point known as a chiasma. So in this image that we can see here, it has it all neatly labeled for us. Those points where the red and blue arms are kind of touching, each of those is a chiasma. So in this image, we have 2 chiasmata shown. And then when this happens, when the blue chromosome trades a piece of information with the red chromosome, we end up with these recombinant chromosomes. And a very, very simplified kind of diagram that I put into these slides here just shows that you start with 1 red and 1 blue. They kind of cross over like that, and then when they kind of pull back apart, you have a red with part of blue and a blue with part of red. And it's important to remember that the parts that they trade have exactly the same kind of information, so it's exactly the same gene, just it may be a different version of the gene. So if the red chromosome has version A of the gene and the blue chromosome has version B of the gene, and they trade that information, the red chromosome now has version B and the blue chromosome now has version A. And then that creates this recombinant chromosome and increases the random combinations that are possible during meiosis. So what exactly is the big deal? Well, meiosis is the thing that makes sexual reproduction possible by going from the diploid cell down to the haploid so that 2 haploid cells can combine to produce another diploid cell that can grow and develop into an organism, and the cycle can start again. If that didn't happen and 2 diploid cells were to merge, you would end up with a 4N organism that may or may not be viable, and probably not going to be viable, especially in organisms like animals. Plants are a little different, but plants are weird and we're not talking about them right now. Let's ignore plants. Plants don't exist right now. And then you get this random shuffling of chromosomes that increases that genetic variation. And when you get this increase in the genetic variation through that random shuffling of the chromosomes and also through that crossing over that furthers the increase in genetic variation, you increase that genetic variation in a population, and that is going to overall make the population more able to adapt to things that their environment may throw at them. So that's basically meiosis in a nutshell. Please feel free to comment any questions down below. I know meiosis is a little bit of a tricky subject, but the important things to remember are where crossing over occurs and how it's different from mitosis, and the big deal that you're going from 1 diploid parent cell ultimately to 4 haploid daughter cells. So I hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.